Good morning, everybody, and welcome. Well, it's 11 a.m. on the East Coast in the United States. That can only mean one thing. It's time for the weekly Fralin Biomedical Research Institute Pioneers in Biomedical Research Seminar Program. And before we getting around to introducing our introducer for today's speaker, Dr. Scott, I want to do as I always do and put in a plug for next week's seminar. Next week, same time, same station, we'll have Dr. Niels Kramer from the Department of Psychiatry in the University of Tübingen in Germany. And Dr. Kramer will be talking about running on dopamine, metabolic regulation of reward seeking. And so I want to remind everybody for the ground rules again, our speaker will be presenting a PowerPoint presentation. And as you have questions, please enter them in the Q&A. And the moderator will handle addressing the questions to the speaker. And so without further ado, it's my great pleasure to introduce Dr. Jenny Munson, who will introduce today's speaker, Dr. Scott, on behalf of the FBRI and the cancer group at the FBRI. Jenny, all yours.
Okay, thanks, Mike. Thanks, everybody, for coming out this morning. It's my pleasure to introduce Dr. Evan Scott, who's an associate professor in biomedical engineering, as well as microbiology and immunology at Northwestern University. Evan got his bachelor's at Brown University and his PhD at Washington University, both in biomedical engineering, after which he did a postdoctoral research fellowship as a Whitaker International Scholar at the Swiss Federal Institute of Technology in Lausanne, Switzerland, which is where we met 11 years ago now, which doesn't feel like it was that long ago, but it is. And since then, he has, starting his professorship at Northwestern, he has won numerous awards, including the NIH New Innovator Award, DP2 mechanism, as many of you may be familiar with, as well as the NSF Career Award. He's also was named one of PRISM's 20 under 40 for the American Society of Engineering Education. And he had both a predoctoral and an early and a scientist development grant with the American Heart Association. He's also won a number of other awards, which I'm not going to have time to list, in the fields of biomedical engineering, nanotechnology, immunology, and biomaterials. He's an amazing innovator in the development of new syntheses and novel materials for immunomodulation. And he really does everything from kind of coming up with the idea, thinking about the immunology, to conducting the animal experiments and kind of figuring everything out, which I think you'll see today in his work. He's also has a number of patents and publications in this area. And he's also just a really great person. and he's mentored a number of students and is heavily involved in education, mentorship, and diversity efforts, both at the national and local levels. And so it's my pleasure to introduce Evan as he talks to us about his nanobiomaterials for immunomodulation. Thanks.
Okay, thank you so much, Jenny, for that introduction. And it's really great to see you again. I hope next time I can revisit or come back and actually see you guys in person. But this is, how things are these days. So I'm going to share the screen really quick and go full screen. Okay. So yes, today I'm going to be talking about some of our recent work in the area of designing nanomaterials to interact with immune cells. So hence, essentially engineering the nano-bio interface between these materials in immune cells in order to enhance immunotherapy. So I am essentially an immuno-engineer. My background is in biomaterial science, but we spent a lot of time during our postdoc, Jenny and I, working in the area of immunology. And so I'm now in this field of combining those two things together. So this is how I divide my lab up. It's in 3 separate areas. First, I'm a biomaterial scientist by training, so we do a lot of nanomaterials work, biomaterials work. We work on hydrogels, self-assembled systems, and then we try and test those to see how they interact with the immune system. And sometimes we design them to interact with very specific cells within the immune system to probe questions. So we do a lot of immunology as well. And finally, we try to combine our new materials with what we learn about the immune system to develop new kinds of therapies. And so we've actually been able to branch out into many different areas. Our main areas were primarily in cardiovascular disease, initially with the development of immunotherapies. And we try and use a technique that we're calling immunotheranostics. So it's essentially theranostics where you can have a nanomaterial that is both diagnostic, so you put a fluorophore, some kind of imaging agent in there, along with the therapeutic. So you can actually see the biodistribution, like where the actual therapeutic is going. And so we find it's really useful for studying immunology because it's really important to figure out which immune cells within this whole network of the immune system are actually being targeted and being manipulated. So we try to both deliver our therapeutics along with some kind of imaging agent to see, what's actually going on. And this allowed us to actually branch out and do more mechanistic studies in these a variety of different areas. So today I'm going to touch upon just a few of the areas that we've been working in and end with a longer discussion of one of our cardiovascular projects. So just to make sure everyone's on the same page, we work with nanomaterials primarily. This is the size range that we're looking at. Let me see if I can get a pointer. There we go. This is the size range we're looking at, around the size of viruses. These are some of the more common types of nanomaterials you'll see. You've probably seen this image before. It's very commonly used by people in drug delivery when we talk about our nanomaterials. We actually don't work with any of these structures. The closest one we work with is micelles, but we mostly work with synthetic polymeric self-assembled systems, and I'll talk about some of the structures that we work with. But I like to use the word nano-biomaterials, primarily because we want to make sure they're compatible, they're biocompatible. And so there are a lot of nanomaterials that actually aren't going to be as compatible. And so that's a big focus for us, is making sure that our materials are going to be interacting very nicely and with the body in a safe way. And so one thing that's kind of overlooked many times is that nanomaterials are actually very common in our bodies all the time. They're involved in many different types of processes. It's a critical part of tumor metastasis, where exosomes are released to modulate draining tissues. It's important for neuron communication. Lipids, of course, are transported through nanoparticles, through HDL, LDL. And so really important for us is that nanomaterials are actually a form of communication that's used within the immune system. They use nanomaterials to communicate between each other, different immune cell population. They can deliver mRNA and different factors. And also immune cells are going to be taking up viruses that are on the nanoscale. So they're very good at taking up nanoscale materials and processing them. And so with that in mind, we focus quite a bit on biomimicry. We try to mimic some of the structures and the functions of many different nanoscale natural things, such as viruses. For us, viruses are really important because they're really good at being taken up by specific cell populations and specific tissues. And so we try to mimic their structure, the way they present their targeting ligands on their surface, controlling the densities on the surface. And this is where the engineering comes in. We can engineer these materials that we work with in terms of their size, their shape, their charge, their surface chemistry, all to try and better mimic how viruses interact with immune cells. And so this has actually become a really critical part of this area of immunotherapy, which of course was the breakthrough of the year many years ago. I believe this was back in 2013. But one thing that we've come to terms with over the last, I guess, 7 years or so, is that it's really important to understand where these immunotherapeutics are going, what immune cells they're actually interacting with, and to better control that. And so as a result, controlled delivery has become a really key part of many platforms going forward. So of course, nanomaterials can load a variety of different molecules, RNA, DNA, small molecule drugs. They can be enhanced or employed for unique strategies for like controlled release and slow release systems that become really important for determining which organs are going to be taking up the particles in the drug and how long they're going to be available systemically. And so we actually do both of these. I'm going to talk a little bit about one of our hydrogel systems for sustained delivery, as well as how we can actually enhance the targeting of our nanomaterials to specific organs and tissues. In particular, we're mostly interested in targeting immune locations where immune cells are going to be found. So secondary lymphoid organs, for example, we like to target our particles to the spleen, the lymph nodes, also to the liver as well in certain situations. And we're mainly targeting these cell types, APCs, antigen-presenting cells. These are primarily going to be dendritic cells, macrophages, monocytes, and B cells. And the favorite one for us is going to be dendritic cells. So this is kind of what a dendritic cell looks like here. You can think of them as kind of like a general that's going to order or give orders out to a military, which is composed primarily of T cells. So they go around, they collect information in the form of viruses, and they process that information and they distribute that or present that to T cells. And now the T cells can know what to do and go out and attack, say, a virus-infected cell or a cancerous cell. And so this is that interface, this bio-interface, nano interface that can actually occur between, in our case, our particles in the cell surface, as well as then you have this other interface between immune cells, different immune cell populations. So interfacial interactions are quite important for immune responses. And this is what we're trying to tap into here. And this is a schematic of a dendritic cell. And what's really amazing for us, useful for us, is that the dendritic cell controls both arms of the immune response. They control the pro-inflammatory response as well as the anti-inflammatory tolerogenic response. And it all depends on how they process information and present that information to the immune system, particularly T cells. Shown up here is just their collection of various things in solution or in the blood, such as pathogens and chemicals. In particular, with like viruses, they can take up the viruses internally, process them, break them down into peptides, present those peptides on the surfaces in complex with specific receptors, primarily MHC, as well as interact with certain receptors like TLRs on their surface and internally that trigger pro-inflammatory responses. And so as a result, you have this MHC peptide complex presentation. You have specific receptors called co-receptors being expressed in response to inflammatory signals, and very specific types of cytokines, mixtures of cytokines being released that give this pro-inflammatory signal. Now, if you actually go the same route, but deliver anti-inflammatory signals, you'll have a different effect. You'll have decreased numbers of these co-stimulatory molecules being expressed on the surface. You'll still have presentation through MHC, but without those co-receptors and with a different repertoire of these cytokines being expressed, you will activate different types of T cells. So in this case, you're reactivating primarily regulatory T cells that inhibit immune responses. And so that means that depending on what we deliver to these dendritic cells and how they process these nanoparticles that we give to them, we can actually shift the immune response to a tolerogenic state, which can be really useful for things like organ transplantation and anti-inflammatory treatments for heart disease and vascular inflammation. Or we can deliver something like an adjuvant for a pro-inflammatory response, which would be really useful for things like cancer immunotherapy and vaccination. So this is what we try to do. We try to mimic the structures of these viruses. We try to mimic their biochemical composition so we can take pieces of these and use these to actually direct the kinds of responses that dendritic cells are going to give to T cells, which is also just the basis of how vaccines essentially function. You have your antigenic target. This is usually a protein. In our case, we also work with antigenic lipids as well as an immunomodulator, most commonly thought of as adjuvants. But in our case, I like to use the term immunomodulator because adjuvants are usually associated with a pro-inflammatory response, but we also deliver modulators that are anti-inflammatory. So I like to use immunomodulator in that case. So these are the two things we're trying to deliver. And these two different types of payloads come in many different shapes, sizes, structures, and ranging from lipids to nucleic acids. And so it's really important for our delivery system to be able to load a variety of different types of molecules. But really importantly, are we going to deliver these molecules to the right location and the right cell type? So how do we control that? So we spend a lot of time making our particles specifically designed to be taken up and endocytosed by these specific immune cell populations. And how this has been done classically is by making particles stealth, which is essentially putting a coating on the particle of a hydrophilic polymer that inhibits a lot of non-specific cellular interactions. And so this was primarily developed many, many years ago. Actually, it was developed over the course of about 17 years, initially with this first nano drug that came on the market, which is Doxil, which is the liposomal form of doxorubicin. It took a really long time to figure out why it wasn't working initially. And it turned out that putting this coating of a hydrophilic molecule on the surface, in this case, PEG, polyethylene glycol, allowed better circulation time throughout the body, allowing the particles to be taken up actually within the tumor at a higher amount. And so this has become a really go-to method in the field. To build upon that, you can put targeting moieties like antibodies on the surface or some kind of peptide ligand that can interact with receptors on the surface of the cell to further enhance that specificity of the uptake. But even with both this PEG coating as well as some kind of targeting ligand to enhance uptake, we still get a lot of non-specific uptake of these nanoparticles. And that is due to this system here, the mononuclear phagocyte system. And this is essentially a system of phagocytic immune cells, inflammatory cells that are located in various organs throughout the body that are evolved to essentially collect these particulates that shouldn't be there in your bloodstream. And it's their job essentially to remove and clear these nanoparticles from circulation. And essentially they still accumulate most of the materials. So it's really important in developing strategies to avoid uptake in these various organs and locations, such as primarily the liver and the spleen are going to be the big ones, but also the kidney and the lung as well. So what are some of the newer strategies that we can use to try and get around this non-specific uptake by these cells? So some things that we're working on in our group, first is taking this targeting moiety that's on the surface of the particles kind of to the next level, which is trying to optimize their density and the mechanism by which they're presented on the surface to better interact with the receptors on the cell surface. So you can actually match, or this is something we do in vitro, is match the presentation density of the targeting ligand with the actual expression density of that receptor on the cell surface to enhance the uptake by a specific cell type. Another thing we can use is the morphology and the shape. It's been shown by us and as well as many other groups that if you change the shape of the actual nanostructure that you're working with, that can strongly impact membrane interactions as well as uptake. And Dennis Disher did a lot of this work, demonstrating that filamentous and elongated structures actually have difficulty being taken up by macrophages and monocyte populations. So these an elongated high aspect ratio particle will actually circulate much, much longer than a spherical particle. And we've actually seen very similar results to what the Disher group has shown. Something we focused on quite a bit more recently, this is a paper we have currently in press, which hopefully will be out in a few weeks or maybe actually probably a month or so, is controlling the protein corona. So when you place a nanoparticle in blood, it's immediately going to absorb onto its surface blood proteins. And so how these proteins actually interact with the material surface can cause them to actually denature and present various ligands and binding sites to the immune system. So if you can actually control the surface chemistry to modify the chemistry, the conformation of proteins, that can actually be a really critical way of controlling where particles go and what cells take them up. So that's something we've recently started working on. And of course, the biomimicry, which I mentioned before, you can actually take certain things that are used by biological systems like CD47, for example, is a don't eat me signal. That's on the surface of red blood cells and many different cells in the body to tell immune cells not to take these particles up or not to eat or destroy red blood cells. So you can actually take CD47, put these on nanoparticle surfaces to better mimic these kinds of signals. And something that we've just submitted, which I'll talk briefly about, is actually just shutting down that mononuclear phagocyte system. So we have a pre-injection strategy where we actually inject a nanoparticle that's designed to be heavily taken up by the liver and the spleen, but it delivers a non-toxic agent. We use latrunculin in this case to inhibit non-specific uptake, primarily macropinocytosis. And so if you do this pre-injection and then follow that pre-injection up with your actual payload that you want to deliver, you can actually see drastically decreased uptake of your second injected nanoparticle by these cells within the spleen and the lymph nodes, allowing this particle to circulate for longer periods of time to actually reach the target of interest. I'm going to talk just briefly about these two, just because we find these really interesting and we like to kind of promote this idea a little bit more. So first is the area of right here, the protein corona. So the protein corona is essentially this layer of proteins that will bind to the surface of materials. And in nanomaterials, it forms these various different kinds of structures. So you can have exposed domains within proteins being exposed on the surface. You can have aggregates of proteins on the surface. But what's interesting is that scavenger receptors on the surfaces of certain immune cells are designed to interact with different conformations of these proteins to take up things that shouldn't be in circulation. So by modifying how these different proteins, their conformation are displayed on the surface of particles, you can actually change interactions with receptors on these cells. So we're wondering, okay, how does these different surface chemistries actually modify the structure of various or common proteins, such as albumin, which is the highest concentration protein in blood. It's a dominant component of the protein corona, especially at early stages of injection of a nanoparticle. How does these different chemistries actually influence the structure of these proteins, and how does that result in differences in cell uptake? So this is a study we just completed where we looked at albumin and its conformation on the surface. And one interesting thing we found was that this phosphate chemistry was actually able to retain and maintain the native conformation of albumin when it bound to the surface, whereas these other really commonly used surface chemistries like methoxy and hydroxyl groups actually denatured albumin quite a bit. And we could do this. This is circular dichroism data looking at the alpha helical change that can occur, and it maintained the alpha helical content of albumin only when we use this phosphate modified surface. And this drastically changed the biodistribution within the mononuclear phagocyte system when we injected these into mice. And so, for example, the phosphate surface actually resulted in decreased uptake in the kidneys and a drastically different uptake in terms of different types of monocyte populations that were taking up these particles. And we found that this was actually a result of the SRA1 scavenger receptor. And so we use inhibitors to it to show that uptake was only inhibited by the hydroxyl and methoxy surfaces, whereas uptake was continued in the presence of this with the phosphate modified surfaces. And so we came to this conclusion here that the mechanism for this was that the phosphate surfaces were actually maintaining the native structure of albumin, whereas these other structures where these other surfaces were actually denaturing albumin, resulting in uptake through interactions with SRA1 scavenger receptor. So we're now investigating a bit more the use of the phosphates and also exploring other surface chemistry to see how this actually modifies uptake within these mononuclear phagocyte cells of the mononuclear phagocyte system. And then briefly, this other newer method we're using to avoid uptake by that system is this inhibition of the MPS. So this is a nanoparticle we designed that's loaded with latrunculin. So latrunculin disrupts the cell cytoskeleton and primarily disrupts macropinocytosis. But it is toxic on its own at high concentrations, but we found that when we deliver it at lower dosages and in the controlled format without nanoparticles, it's no longer toxic. We've actually used this. I'll talk about one project where we use this for at the end. It's really important for our treatment of glaucoma. I'll explain why that's the case later on. But we've already used these in mice and shown that they're non-toxic. So we know that's the case. But what's really interesting here is that you can actually cause selective inhibition of macropinocytosis while still allowing receptor-mediated endocytosis. So this means that we can actually put a targeting moiety on these particles, load them with latrunculin, or have this pre-injection of latrunculin loaded particles to inhibit. This is a case here where we're inhibiting macropinocytosis, but not inhibit receptor-mediated endocytosis. And so this is kind of the strategy of how this would actually look. So say you have a tumor in a mouse, you have a pre-injection of these inhibitory particles. We call these MINPs or MINPs. You inject those, they shut down the mononuclear phagocyte system, and then you can follow this up with a second injection of a receptor-targeted nanoparticle. In this case, we're using something that targets folate because it's found in many different types of tumors. And in this model, we're using a B16F10 model, tumor model, which expresses folate or folate receptors. So we have this second injection. And in the case where you inhibit the MPS, this particle can now circulate better and be taken up more easily inside of the tumor. Whereas without this pre-injection, this particle will circulate and be taken up primarily within the liver and the spleen like normal, and you have less available to be taken up by the tumor. So we spent a lot of time trying to optimize these different injections. We found that the best system actually worked with a pre-injection about 4 hours or 24 hours ahead of time. So we tested 24 hours ahead and many different time points before. We're doing a pre-injection 24 hours ahead of time, and then following that up with a co-injection of the inhibitory particle with our key therapeutic particle at the same time, resulted in inhibition of uptake in the spleen and liver of these particles, while allowing circulation to increase for the targeting particle. And so within the tumor microenvironments, we saw drastic increases, up to 8-fold increase in uptake by the targeted therapeutic particle, just because we had this pre-injection of this inhibitory particle ahead of time that decreased uptake in the spleen and the liver. So this is something we have currently. It's in preprint now, and it's currently under review. So that's our strategies that we're currently working on for enhancing targeting of our materials, but I haven't talked about the kind of materials that we're working with. And so this next section, I'd like to discuss some of the key platforms that we focus on and why that's the case. So we primarily work with self-assembling amphiphilic molecules. So they have a hydrophobic side and a hydrophilic side, and they self-assemble to a variety of different kinds of structures that are great platforms as nanobiomaterials. But I want to preface this by talking about some issues in the field, because I like to be very honest and straightforward, because I think this has been actually holding back nano delivery a little bit. Is that sometimes people get very excited, when you have a new technology and you go full force ahead, and you sometimes overlook a few things that are really critical for optimizing how these things work. So currently we're at the stage where there are a lot of nanotherapies being tested, but not a lot are actually getting to the market. And some things that are inhibiting this is the fact that the majority of the particles are still not getting to the area of interest. So there's a lot of work done by Warren Chan recently publishing work showing that about 90% on average of your administered nanoparticle actually goes to liver and spleen, and less than 3% is actually getting, say, for example, to the tumor that you're trying to target. It's actually pretty amazing that 3% actually has such a potent effect, but you can't ignore the fact that 90% is not getting there. We're also kind of plagued by the fact that many of our strategies are just really complex and they're difficult to scale chemistries and fabrication methods. We still have issues with loading different kinds of molecules. In particular, biologics like proteins are extremely difficult to load inside of nanoparticles at high levels. And anytime we try to load more than one type of molecule, particularly if the molecules are different, like loading a lipids plus a protein plus a small molecule together in the same particle, you get differences in uptake for each one of those, or differences in encapsulation efficiency for each one of those, making it very difficult to have reproducible formulations when you have more than one type of payload. But when you do it right, there are just so many benefits to using nanotechnology that it's really worth this extra effort that we're putting into it. You can decrease this non-specific uptake by using strategies like some of the ones I mentioned before. You can target drugs, specific cells and tissues, drastically lower, this is really important, drastically lower the effective doses that you have to administer. We have cases where we've lowered the dosage by over 600-fold just because we targeted where the drug went and enhanced the ability of the drug to go into cells. And I'll give a quick example of that later on. You can repurpose drugs. So maybe one drug works a certain way when it's delivered systemically, but if you actually change the biodistribution in the cells that take it up, you can actually repurpose it and have it actually function through a completely different mechanism. That's one advantage of nanomaterials. You can completely change that therapeutic effect, and you can do this, of course, theranostic strategy where you can see where the drug is actually going. You can see the nanoparticle, so you can see what cells are actually taking up your drug to better understand the effect that you're seeing. But I really want to highlight the fact that these mononuclear phagocyte systems, they are a critical part of the innate immune system. And as a result, nanomaterials are actually quite good at modifying immune responses. They're actually, this is probably one of the best strategies for them, is to actually use them for immunotherapy, because we have this evolved system with APCs where they're actually taken up more efficiently. And so this is actually where we try to use our strategies. We try to take advantage of this to develop different kinds of immunotherapies by targeting these APCs. So these are the systems we work with. We work with self-assembled systems. And this is the block copolymer we work with. It's a polyethylene glycol block. So this is the PEG block. And we use polypropylene sulfide as the other block. So this is actually extremely hydrophobic polymer, whereas PEG, of course, is very hydrophilic. You link these two together, have a water-soluble side, a water-insoluble side, and depending on the volumetric ratio of these two, you can get different kinds of curvature in the structures when they self-assemble. And this is very, of course, exactly what happens when you have self-assembled phospholipids for making cell membranes. It's the exact same strategy, except we try to control this with synthetic polymers to get all kinds of different types of shapes and structures. And we optimize this in vitro just to see what kind of shapes and structures you'll get. So as you change the chemistry, so for example, if you switch out the hydrophilic block for something else, you're actually going to get differences in the thermodynamics of the system. You're going to get different kinds of structures forming. And so Molly in my group has been working quite a bit on how you optimize these systems. She developed her own new block copolymer system and showed how it changes the conformation when you change the hydrophilic block, and how we have to go through and reassess what all the different ratios should be to determine what kind of structure you'll get. Because as you change the ratio of the hydrophilic and hydrophobic content, you will change the volumetric ratio of the structure in terms of the hydrophilic and hydrophobic blocks, and you'll get a different kinds of structure. So this is a publication we had recently demonstrating how you can have a high throughput optimized assessment of these new types of block copolymer systems. For us, really critically, as I mentioned, one strategy that we have for targeting is to use the shape and size of these particles. And so if you have the exact same surface chemistry, this is actually an advantage of using self-assembled systems, is that you can have the same surface chemistry. All of these particles have the same peg on the outside, but they have different shapes. It's a nice way to eliminate variables you get by changing the surface chemistry. If you want to try and get a different structure by changing the chemistry of the polymer, you have now different protein absorption. In our case, we have the exact same chemistry, but we can make these different shapes. And so a lot of our studies have shown by injecting these different shapes and structures, you can get drastically different uptake by different immune cell populations. So this is looking at, I wish I had more time to talk about, this is a really kind of unique structure I really like working with called a bicontinuous nanosphere, shown here. It has a cubic lattice inside. I just don't have time to talk about it today, but we do have several publications on it that we're trying to promote this as a delivery vehicle. But it has these channels going through it, and it has a larger size, and it has a very different biodistribution than all the other particle types that we've looked at. For example, like filaments, which are just not taken up very easily by anything, versus micelles, which love to be taken up by almost everything, and polymersomes, which have this kind of intermediary uptake where they're actually preferred to be taken up by dendritic cells. And so we've shown this in many different situations. This is a sub-Q injection where we're developing a vaccine, looking at the different uptake of these different kinds of nanoparticle shapes. So as a result, using this PEG PPS system, we've made a variety of different kind of nanostructures and used them for all kinds of different kinds of applications. So I'm going to talk today briefly about one application that we've used using these filamentous structures we thought was rather unique. And I'm going to touch upon our newer system, this nanogel system here, as well as one of our polymersome systems that we've been using for delivery for heart disease. So one thing I want to point out about this PEG PPS system is that the polypropylene sulfide block, although it's very hydrophobic initially, it oxidizes into a hydrophilic variant. And so by oxidizing it, you shift that ratio of the hydrophobic and hydrophilic. And so you can actually use that as a trigger to cause the particle to denature and fall apart. You're actually disrupting the thermodynamics of the system by making this more hydrophilic. And there's now a new optimal conformation. And so it will reorganize into a different structure. And when that happens, the payload will come out. So these particles are oxidation sensitive, and you can use this oxidation to trigger the drug release. And very recently, we've actually found that using the fully oxidized form of PPS, this PPSU, polypropylene sulfone, actually has a lot of advantages. And that's the basis for this newer system I'll talk briefly about called our nanogel system. So here's some of the advantages of using the PEG PPS system. We found that there's actually a scalable method of making these different kinds of structures. Anyone who's worked with liposomes has probably worked with this method called thin film rehydration, where you dry a film of lipid on the surface of a glass container and you shake it in water, and it will assemble particles primarily randomly. It's not very high throughput, it's difficult to work with, and it's not very scalable. And so one of the first things I started working on in my group was developing a high throughput method. So this method of flash nanoprecipitation, it's a mixing chamber essentially that at microsecond timescales will mix organic in an aqueous phase to cause phase separation very reproducibly and nanoprecipitation of these particles. It allows us to make all these different structures very quickly and at high throughput. So we can use this to make gram scale amounts of these nanoparticles. So we're not limited for that going forward with higher animal studies. And so we studied this system in primates and found that the targeting is very similar in mice as in primates. It's also non-toxic in primates, so we were quite happy to find that out. So you don't want to spend a whole lot of time optimizing something in mice, and then you have a complement response or something toxic later in higher order mammal. We focus quite a bit on encapsulation efficiency. So we have a system now that was just published that can actually load almost any molecule that we throw at it and any combination of molecules at greater than 96% loading efficiency. So I'll talk briefly about that system. The PEG PPS system is quite good at controlling that structure that you form, which allows us to get these very specific targeting, of organs and cells. the oxidation sensitivity allows very nice release into the cytosol of cells. So this is a release from the endosome, so endosomal, within the endosomal pathway. So releasing from the endosome into the cytosol upon light triggering. So we can use like photooxidation to control this. This is actually something I did when I was in my postdoc with Jenny in the Hubble group. And one really nice thing about the PPS system is that it's non-inflammatory. So many different nanoparticle systems have a background immunostimulation. So for example, PLGA, if you just inject naked PLGA with nothing in it, it will have some kind of inflammatory response. Depending on the surface chemistry, sometimes that's anti-inflammatory, in other cases it's pro-inflammatory, which makes things a bit more complicated. But we did a lot of work, RNA-seq analysis, to look at what exactly going on with these different cell populations when they take up PEG PPS. And it's actually a very, very minimal change relative to other nanostructure platforms. And so for us, that's advantageous because the response you get is dependent on the payload, which allows customization, and it's not so much influenced by the actual delivery system itself. And so doing that, we can actually decrease side effects quite a bit for a variety of different kinds of drugs that we've worked with. So first, I want to go in a little bit more detail about one advantage of using the PEG PPS system for sustained nanomaterial delivery. So nanomaterial delivery has this, I guess, standard method of administration, which is to do bolus injection, IV typically, into mice or to the patient. And so if you want this to happen over longer periods of time, you just do more injections. There are actually very few platforms for slow release of nanoparticles. We have platforms for slow release of drugs, but not so many for slow release of the nanoparticles. So you don't have to keep repeating these injections. So this is one thing we actually wanted to do, was develop a slow release system for nanoparticles. And so something interesting about the PEG PPS system is that you can make these filaments, but this is not actually the thermodynamically most stable conformation. And so over time, it will actually reorganize itself into something that's more stable. In this case, switching from this high aspect ratio structure to a micellar, a spherical structure, which is more stable. This actually happens depending on the chemistry of the system over the course of weeks to months. And we started seeing this in our formulations, particularly when they're exposed to air. So this is actually enhanced by oxidation. And I'll talk a little bit about that, how we know that a little bit later. Well, you can actually see at the ends of the filaments, these locations where the micelles actually kind of bleb off of the ends and switch over to this different nanostructure. We looked at this in a lot of detail. We did like 3-dimensional tomography at the nanoscale to see these different blebs of micelles coming off, and it's very uniform. The release of these particles, you release very uniform micelles at the ends of these filaments. So we wanted to find a way to actually take advantage of this. And so we cross-link these filaments into hydrogels. And you can load drugs into these filaments and make these gels essentially drug depots that will essentially release these nanoparticles over a long period of time as the hydrogel degrades. And so we've done quite a bit of work. This is one of our main platforms, this hydrogel platform that's injectable. So this can be an injectable formulation, initially liquid. You inject it into the mice and they form a gel in place that has this delivery system. So making this work required us to make a heterobifunctional polymer that has one end that can be cross-linked together. In this case, we're using a vinyl sulfone group to do this. This work was mostly done by my student, Nick, who recently graduated last year. He's now working in industry, but he did a lot of chemistry to get this to work. And we can mix together these different blocks to get different kinds of chemistries displayed on the surface. And one is this cross-linking moiety. And by doing this, we actually have found it's kind of a new form of hydrogel degradation. So classically, you have these two different methods. You have surface erosion, where the gels will degrade from the outside in, layer by layer, essentially. Or you can have a bulk degradation, where everything is breaking apart all throughout at the same time. You get these usually non-uniform chunks coming off of the part of the hydrogel as it breaks apart. In this case, you're breaking it down into metabolites. So you have like individual small molecules and monomers coming out. In our case, we're actually degrading the hydrogels into monodisperse nanoparticles. So they're not breaking apart into random structures. They're not breaking apart into individual monomers. They're actually breaking apart only into monodisperse nanoparticles, which we can take advantage of. So this is what these gels look like. You can cross-link them together into different kinds of structures, depending on like if you use a mold. This is just a a spherical mold. You can load cells inside them. You can load other nanoparticles inside of them as well. And they always seem to have a really nice, independent of the actual cross-linking density, a really nice monodisperse nanoparticle population being released, these micelles here. And this is because it's all thermodynamically driven. This is work that we did modeling this system to figure out how this actually is working. And it turns out to all be dependent on interfacial tension between the PPS components and the aqueous solution. So we can measure interfacial tension in collaboration with Professor Ken Scholl. He's in the material science department at Northwestern. And we modeled these self-assembled systems using essentially the classical thermodynamic modeling for nanoparticles, where you have this elastic hydrophobic interior that has a certain elastic energy that doesn't want to stretch out too far, because if it stretches out too far, you have the hydrophobic part interacting more with the hydrophilic aqueous solvent. You have steric stabilization of a hydrophilic chain on the outside, and you have the interfacial tension between the hydrophobic components and the aqueous exterior. So you can model those three forms of free energy and figure out what kind of structure you'll get under different solvent conditions. And so if you compare a spherical to a cylindrical structure, this is the equilibrium radius that you'll see at the lowest energy state when it's self-assembled. The cylinder always has a smaller radius compared to the sphere at different surface tensions. And so what this means is that if you actually have a system where you're switching the surface chemistry to change the interfacial tension, you can actually have at higher surface tension this more stable, smaller spherical structure for the cylinder, which minimizes the chain stretching, and it minimizes interfacial tension. So it has the smaller structure preferred at higher tensions. But as you decrease that by oxidizing the particle, you actually go to a phase where you can actually have a bigger interior. You can have a little bit more chain stretching because it's more oxidized, not as hydrophobic. And this allows equilibrium to be maintained by the outside hydrophilic polymer. So it's now steric stabilization controlled, and you can get a bigger radius. And so that allows you to switch from the smaller radius of the cylinder to the larger radius of a sphere. And so it's all thermodynamically driven, and we did a lot of studies looking at interfacial tension to figure out like how much oxidation is required to cause this to occur. So we have a nice mechanism how this is actually working. And so you can load these different block copolymers depending on their chemistry. You can have a fluorophore attached, you could have this cross-linking group attached, but that's the beauty of self-assembly. You throw all of that together, it'll all assemble together into a structure. So we can make these really nice and to be honest, this is a little bit overly complex because we just wanted to get this paper out. This is probably an overly complex system, but this allows you to trace where the polymer is going. It allows you to trace where a model payload is going, and it allows you to analyze this through two different fluorophores. So we can look at flow cytometry to see cell uptake, and we can do near infrared analysis to look at organ level uptake. So we cross-linked this together into our hydrogel that was injected into mice, and we looked at the release of these nanoparticles over time, and we found that they actually release over the course in a controllable mechanism. So we can control this for up to 2 months of slow release of nanoparticles in vivo. We did a lot of work to trace, where are the particles going? Are they intact? Are they actually staying in the micellar form? So we could trace the polymer and we can trace the payload. And we see that they actually go together when they're taken up by cells. So the payload and the polymer are getting to cells at the same time, telling us that they're actually intact in a nanoparticle form when they're taken up by immune cells. So we're currently investigating this system right now for a lot of different applications for slow release of nanoparticles. One example is we can actually inject these as a paratumoral hydrogel that can actually result in slow and constant uptake of myeloid cells within inside of a tumor microenvironment. So we have a grant on this right now that we're working with collaborators at the medical school, Professor Ben Zhang. So this other system I want to talk about briefly is what we've developed to solve this problem of loading efficiency. So if you actually oxidize polypropylene sulfide completely to the sulfone derivative, so this is a polypropylene sulfone polymer. It's not a block copolymer. It's just a single polypropylene sulfone homopolymer. It can actually self-assemble. So this is actually pretty rare. You don't really see a lot of controlled self-assembly of homopolymers because you need that hydrophilic block to control the structure that forms. But we found that this actually is working through a unique form of self-assembly using sulfone-sulfone bonding. So what we found is if you place this in DMSO, which is actually the perfect solvent for this, if you look at the structure, it's very similar to DMSO. It's like a polymerized chain of DMSO essentially. It's very solubilized in DMSO. You'll see an extended conformation. So we did lots of modeling. This is with Monica de la Cruz's group in the material science department. And this work was done by my postdoc, Fan. So it's an extended conformation in DMSO. You place this in water and they start to take on these really unique aggregates. And it turns out that these aggregates were a result of sulfone-sulfone bonding due to a changing in the polarity of the solvent. So in this area, in DMSO, you actually have a lot of repulsion from the oxygen interactions here. But when you increase the polarity of the solvent, like going to water, you actually have screening of some of these interactions. And this allows actually that the dipole across the sulfone group to actually work in terms of bonding. So it's actually similar to like hydrogen bonding. It's a sulfone-sulfone bond that can occur. If you have enough of these, you can actually have very stable aggregates. And it was forming these amphiphilic nano assemblies. So we actually were having kind of just like an amphiphile, except it has to be assembled first from the homopolymer. So we have these crystalline components that are assembled from sulfone-sulfone bonding. And it turns out that as it's forming these structures, the polymer has to fold back on itself to make these bonds occur. And depending on the persistence length of the polymer, it can only fold so much. So at the end, it actually gets to a stage where so much is folded over that this can't actually fold over anymore to get to form these sulfone bonds. And you end up with a hydrophilic sulfone tail. And this results in the ability to self-assemble structures based off of this nano assembly, this nano amphiphile. We're calling it a supramolecular amphiphile as opposed to just using a block copolymer. And so this PPSU is actually very simple to synthesize. You essentially have a very simple protocol also. You just have to have a DMSO solution and add water to it, and it will form these nanostructures. So it forms nanostructures through a unique process that we call network self-assembly. We found out very early on that it was actually forming networks or interconnected networks of these amphiphiles throughout the solution. So we can form a gel. We can actually form an entire gel of the solution using these. But depending on how you add water to the system, you can cause this gel to collapse into nanoparticles. And we found that as this network collapses, it collects anything indiscriminately in the solution around it. So it will just take up any kind of payload. It'll take up nanoparticles as well. We've done actually iron oxide nanoparticles are taken up into these as well. They'll take up DNA, RNA, proteins, and you can actually make a system that has more protein in it than polymer, which is pretty rare for a self-assembled system. But everything loads at greater than 90%. Efficiency. And this is just how it looks. Say if you have fluorescent dextran in solution, you can just add the polymer to this, shake it, and all of the dextran will be collected as you add water to the system, and you can centrifuge down and have all the dextran just inside the particles at greater than 96% efficiency. We can form a variety of different structures with these. We can form filaments. This is the vesicle we can form. These are micelles that we form. And now we've just started. This just came out last month, so we're actually still optimizing in vivo applications. So we've done some biodistribution studies. It has typical size-dependent distributions, but really importantly, we can load mixtures of different molecules. So you can have a protein, DNA, you can have a lipid, and you can have a small molecule. This is a vaccine formulation. Throw all of that together into the same solution, shake it, and it all goes into the particles, each part at greater than 96% efficiency. And so that lets us, mix and match different concentrations of each component in order to optimize the formulation. So we see this as being very useful, particularly for vaccine development, where you have all these different kinds of adjuvants you can mix in to get better control of the immune response. And you don't have to worry about, is DNA going to load better than the protein? Do I have to re-optimize the system because now I have a negatively charged CPG in there, mixed in with a lipid as well? It doesn't matter. It all loads into the particles at the same efficiency, and it all is bioactive, so they release inside of cells, and you can still have vaccine responses. This is an adjuvant response for NF-kappa B activation of macrophages. So we're pretty excited about that system, and we're currently exploring funding sources now to use this as a rapid vaccine development platform. So I just want to have this last section here in the last, I believe, 10 minutes, just to talk about some of the applications that we've used these materials for. One take-home message for us is we like to repurpose drugs. So we tend to use drugs that aren't very exciting. So we have like vitamin D or or rapamycin. Things are used really commonly, but by repurposing them, by changing where they go inside the body in terms of their biodistribution using nanoparticles. And so one of the first things we worked on, started out in my postdoc, was actually vaccine design. So we started out working with how do you make vaccines that work better in infants, so neonates, have different immune responses, particularly with their dendritic cells, relative to adults. And so we worked quite a bit with Boston Children's Hospital. They have an adjuvant discovery center where we can load different types of new adjuvants into our systems that are very stable. This is our particles over a course of 180 days, no change in structure. When I was actually doing my postdoc, I started this project and I had to leave to start my lab before I'd finished this. And I wasn't able to pick up this project again for 3 years. And so I recontacted my collaborators at Boston Children's Hospital and said, hey, would you like to finish this project? And they said, yeah, we actually still have some of your particles from 3 years ago. Should we still use them? And it turned out they worked the same after 3 years. And so we're really happy with the stability of these particles. That comes with using a polymeric system. They're just way more stable than lipid systems. And so these vaccines were stable at 4C for 3 years. We don't have publication on that because it wasn't controlled, but in this study, that's essentially what happened. And so we can load different components into these, to the hydrophobic membrane, as well as load protein inside the interior. And it was really nice in terms of mimicking other kinds of attenuated vaccines, like vaccines that have the entire pathogen present. We can mimic the structure, and we can also, in this case, mimic the composition of BCG, which is for tuberculosis. So tuberculosis has actually become something that we've worked in a bit more after working on that initial vaccine. This is my collaborator here, Chung Wu. She looks at different types of T cells that actually respond to lipid antigens. So bacteria actually have very conserved lipids within them, like macolic acid, for example, is found in Mycobacterium within their walls. And so it's very common in these bacteria, but it's not found naturally in humans. And so we actually have evolved T cells that respond very specifically to different types of lipid antigens. So you think a lot about antigens for T cells, you think of peptides. There's actually lipids as well that can function. It's just not CD4s and CD8s. These are called CD1 restricted T cells. And we actually have 5 different variants of these, and they're very much understudied and haven't been really incorporated into vaccine formulations. So we've developed a nano delivery system to load these different types of lipid antigens. And these can be administered intratracheally. We did intranasal, intratracheal, IV, sub-Q, and we found intratracheal was the best, especially for tuberculosis. We have very nice uptake in the lung. We can have proliferation of these specific subsets of T cells. And so we can only do this because Chung Wu actually developed humanized mouse models that have all these different T cells in them. Something that's very interesting is that these 5 different variants of T cells are found in humans, but mice only have 1. These are called NKT cells, which you probably heard a lot about, but they don't have the other variants. And so it's really hard to study some of these platforms. And so this is done a lot due to the fact that we have a collaboration with Professor Wang. Another area we're looking into currently is Chagas disease. This is a newer project we have. We're just finishing up this paper now. I'd like to bring more attention to this disease because we think this is actually something that could be spreading in the US in coming years. Essentially, there's a parasite, Trypanosoma cruzi, that's actually found inside of this bug here, a kissing bug. It's prevalent throughout South America, and this is a big problem in South America because the parasite actually causes heart failure. It induces inflammation of the heart and is one of the leading causes of heart failure in South America. And typically because of the climate, this bug lives primarily in South America, but it is now actually spreading to the US due to changes in the climate. We actually just found this bug a few years ago in Chicago, and you know it's cold here, and we're seeing this bug actually up here. So it's spreading across the US currently, and it's estimated to be several 100,000 people that already have contracted this disease that most probably don't even know it yet at this stage. So we have a collaboration with David Engman, who's been working with this parasite for quite a bit of time. And there's many drugs. There's only a few drugs. They do work, but they are very toxic given at higher dosages. And they haven't been optimized, to be honest, in a very long time. So one of the main drugs is benznidazole. It actually has side effects that are quite severe, limiting their the amount that can given, and it is not very good at actually getting into cells, which is actually where the parasite is residing. So it seemed very straightforward to us to load this into a particle, enhance delivery of the drug into cells, and maybe you can kill the parasites a little bit better. And this actually worked way better than we expected. So because benzimidazole works great for killing these parasites. And so here you can see actually the polymersomes we loaded these into. You can see the polymersomes inside of cells along with the T. cruzi in the same cell. So we did a lot of parasitemia studies in mice. So you infect the mice with the parasites, and you'll see the blood phase of the parasites. Then as they accumulate inside of cells, this goes back down, and then the parasite will change to a different structure or a different form. And then you have a blood phase again, where it actually can go to the heart primarily. And so if you can avoid the second phase in the blood, where it actually goes to the heart, you can actually decrease a lot of the side effects, a lot of the pathology of this disease. And so we found that we can actually decrease the amount of dosage by up to 660-fold. And still get the same efficacy as the drug in free form in terms of inhibiting inflammation of the heart. So this is heart tissue. This is an untreated group. You see inflammatory cells infiltrating the heart. In our case, it looked like normal tissue at these time points. And it was mainly because we were inhibiting the parasite from actually going back to his bloodborne form to transform to go to the heart. And so we're pretty excited about this because it's really hasn't been looked at too much. And we can essentially decrease the dosage of this toxic drug quite a bit. I'll point out this work was done by a postdoc, Sidja, who unfortunately was stolen from me by Merck. Merck started hiring people extensively this year for vaccine development, and they essentially couldn't match their pay. So she's gone now, but I'm happy for her. She's in a good place. And so this last thing I want to talk about is one of our big projects that we just published on last year. We've been working on this for many years, developing targeted strategies for inhibiting vascular inflammation. And so in this case, we're trying to target inflammation that happens within inside of the vasculature in terms of plaques that can accumulate in the walls of vessels. Many people have focused a lot on macrophages. There's a lot of macrophages in here, but it turns out that dendritic cells are actually pretty important for this process as well. So we want to selectively target dendritic cells, even though there's a ton of macrophages there. And dendritic cells and macrophages share a lot of receptors. And so we needed a system that could actually target those selectively. And this is like a nice demonstration of our targeting strategy. We had shown in a previous publication, this spherical morphology with our PEG-PPS system was quite efficient at being taken up by dendritic cells. And we wanted to enhance that further by using a targeting moiety, as well as load inside something that can have this anti-inflammatory response. So it turns out that you have a lot of pro-inflammatory responses due to antigens from HDL and LDL inside of these plaques. So we have an antigen that we took from phospholipids within cholesterol. This peptide, this ApoB100 peptide here. We have an immunomodulator. In this case, we're just using very simple vitamin D. Turns out vitamin D does not get into cells very easily, but when it does, it is extremely potent. And this is that pathway here. It has a vitamin D receptor, which is actually able to inhibit inflammation, inhibit NF-kappa B activation very potently, but it just has to get into cells at high enough amount. So it's very similar to what we saw with benzimidazole. We just had to enhance the intracellular delivery of vitamin D to get a really potent effect. And we use a targeting strategy shown here. And this is something we're continuing to use quite a bit. We have a lipid tail, which allows insertion into the hydrophobic membrane of the self-assembled nanoparticle. We have a spacer, which allows control over how far the targeting ligand is away from the surface. That actually turned to be quite important for interacting with receptors. And also we have the targeting ligand. So in this case, we like to use peptides because they're small. So this construct here allows us to optimize both the spacer, so the spacing from the surface, and the density. So we can load a lot of this at different amounts into the surface of the particle. And so we can optimize this system to discriminate between two different cell types, even if the cells share a same receptor. So for example, macrophages and dendritic cells both express CD11C, but they express it at different levels. Dendritic cells are CD11C high, macrophages tend to be very low in their CD11C expression. So we can optimize this system in vitro to find the sweet spot that you have the optimal spacer length and the optimal density to have rapid enhanced uptake by dendritic cells, but not and enhanced uptake by macrophages. And so this actually worked in vitro, and this is showing that it works in vivo as well. The main take-home message here is that inside the plaque, as well as inside the spleen and lymph nodes, we're seeing enhanced uptake on a per cell basis of the particles, with no real change in the amount that's going to be taken up by macrophages. And so, again, I like to be very honest in terms of nano delivery. You're not going to get rid of completely this background macrophage uptake, but you can enhance the uptake by other cell types. But macrophages, they're just going to take these up indiscriminately. We like to follow these up with our new strategy where we inhibit macrophages going forward, but this was done before we had that platform. Well, you're just not going to get rid of this background. So our focus was really to focus on enhancing the uptake by these dendritic cells. And it turned to work out pretty well. So we can actually activate a tolerogenic state of dendritic cells where they have low levels of CD80, CD86 expression. They still have decreased expression of MHC2. They have decreased expression of pro-inflammatory IL-12, enhanced expression of anti-inflammatory cytokines like IL-10, and we have, very importantly, expansion of regulatory T cells, which are very good at inhibiting inflammation without having a lot of systemic toxicity or side effects. And so we did a lot of analysis looking at these plaques. You can see drastic decreases in the amount of a plaque accumulation. We deliver these particles to mice over the course of 8 weeks. We did a lot of quantitative analysis looking at decreases in inflammatory cells, as well as decreases in lipid accumulation inside of the vessels. And we've now started working on the controlled delivery systems for this. So instead of just the bolus injection, we're now incorporating vitamin D. This is a recent paper, a smaller paper from us, showing you can actually control the release of these micells that are loaded with vitamin D, and we can maintain high levels of regulatory T cells within mice, essentially for up to 2 months at a time, just using these single injection of these hydrogel systems. You don't have to keep coming back. And so I'm out of time, so I'm just going to briefly mention here that we have, this is something very excited about. So it's currently under review. This might take a year or two. I'm not sure how long it's going to take, but we're able to actually rationally re-engineer the mechanism of a drug, in this case, rapamycin, which is used to inhibit T cells directly. And so we decided, how about we just not directly inhibit T cells? We go through a completely different route or mechanism where we do something called a co-stimulation blockade. And this is where we decrease extensively the co-receptor expression on dendritic cells and APC populations, as shown here at the bottom. And it allowed us to actually do islet transplantation into mice with only a few injections. We can maintain normal glycemia in a diabetic mouse using an islet transplantation. We decrease extensively the dosage of rapamycin we have to give. And so we're currently actually exploring many different uses of this rapamycin delivery system. And it seems very simple at first because it's just rapamycin, but it's really not functioning the way rapamycin typically works. And by going a different routes, we can decrease the dosage and have a different side effects or no side effects in this case. So we're excited about this. We have a preprint for this that's currently out, so we can see that while it's still being reviewed. And the very last thing I'll talk about is this is just showing the future applications for this targeting strategy that we have. It didn't work just in heart disease. We've actually modified this for several years for glaucoma treatment. We can load these moieties inside of the membrane of particles, and this is a different targeting peptide. It targets FLT4, which is VEGF R3. It's found mostly expressed on lymphatic cells. So this allowed us to actually selectively target cells of lymphatic background within the eye. It turns out these are really important for glaucoma. Within the Schlemm's canal, this is actually where the fluid exits the eye. And these cells, actually, one of our collaborators found, become very stiff in the case of glaucoma. And latrunculin will soften cells, the cell softening agent, by disrupting the cytoskeleton. And so we can selectively have uptake just by the epithelium, the endothelium within the Schlemm's canal, and not the corneal epithelium, where you actually have a lot of side effects. So this is a comparison between different endothelial cell types, showing the Schlemm's canal cells selectively take up these particles because they are of lymphatic origin. And so this actually works quite well in the mouse model. We can actually decrease intraocular pressure. And so this paper actually just came out recently, and we have a follow-up paper that we're working on. So I will stop there. And I hope I was able to give an overview of the PEG PPS system, as well as our newer PPSU oxidized PPS system. This showing this is a very nice scalable platform, low toxicity, works in mice, not in humans yet, but in primates at least we know that's the case. Very easy to load a lot of different molecules inside. We've hopefully addressed several limitations that other nanoparticle systems have seen. And so we're currently applying them to many different areas. I try to touch upon a few. We actually just got into endometriosis and mastocytosis as well, and glaucoma and Chagas disease are some of our newer application areas. And so with that, I will thank our funding sources, as well as all the students that did, of course, all this work, as well as our collaborators that allow us to do all these in vivo experiments without having to pay for all the mice ourselves. So I'd be very happy to stop there and take any questions that you may have.
Hi, Evan. Thanks for the great talk. In interest of time, I think I'll just do a couple questions. So Joseph Owens wants to know, with the capability for PPSO nanoparticles to uptake multiple analytes, has there been any research looking into the use of these nanoparticles to deliver patient-specific cocktails, such as with chemotherapy, to have less negative side effects or lower potential dosages?
Yes, that's actually an interest of ours, is personalized nanomedicine. So we actually have a grant under review now for a strategy just like that. And yes, you're exactly right. We can actually customize these specifically for a patient because we can load so many different things inside of them. You can have a diagnostic in there along with different amounts of drugs and a very controllable platform. So yes, you're exactly right. And we're working on that currently.
Great, thanks. And then just one more question. So Rob Gordy wants to know, have you studied the effects of repeated administrations of the nanoparticles? And does an initial priming result in sensitized clearance of subsequently administered doses of nanoparticles?
That is a really good question. And that is actually one reason why we like synthetic systems as opposed to a virus or viral systems, which actually can have this background clearance that can happen after, say, the second administration. You might have antibodies against that viral particle. With a synthetic system, we can control that surface chemistry much more. So you will see some effect in terms of clearance if you have an antibody response against PEG. And so that is something that we have that has come up in the literature quite a bit, is that people can have antibody responses against the polymer itself, which is PEG. And we actually see this quite a bit more in women than men because of the actual use of, say, cosmetics. Women actually get exposed to PEG a lot more than men do. And so a lot of patients have these background antibodies against the particles, which can result in heightened clearance upon multiple administrations. And so that's something we're looking at now. How do you avoid this effect? To avoid this, sometimes we can switch the surface chemistry. We can use a different hydrophilic polymer. We can change to use that phosphate chemistry. So we saw that effect less using the phosphate chemistry. But yes, that is actually something that happens. It happens much less with synthetic systems than it does with, say, a protein-based or a viral system. But yeah, that's a very good question. Yeah.
Great. And just one last question. Samantha Howerton asks, what's the oxidizing agent in the PEG PPS system, such as if you're in a hypoxic tumor microenvironment? Might you see a different degradation?
Yes, you will. We use hydrogen peroxide because it's pretty simple to use outside the body. We also use photooxidizers to do this as well. But in the body, any reactive oxygen species, singlet oxygen species that are present when you have activated inflammatory cells, those are the main culprit here. So yes, you will see an effect from activated inflammatory cells due to reactive oxygen species release.
Great. So to be cognizant of time, and Evan has the schedule to meet with other people as well, I want to thank Evan for giving us a great talk on an overview of all of the different interesting biomaterials he has for a number of different applications. And thanks everybody for attending.
Thank you.